Welcome. This is Jen Bayer with USGS and PNAP. If you don't know me, I'm the lead staff for PNAP. I am happy to be here today with colleagues from Columbia Basin Research and Mari Williams from Pacific States Marine Fishery Commission StreamNet project. We're here presenting the fourth webinar in a 5-part series focused on learning all about data and tools and applications and various things that might help our community practice with respect to pit tag information. So hope you've been following along for the last 4 weeks. If you've missed out, you can find previous presentations on the PNAP YouTube channel. And before I get into today's session, I want to remind you that there's one more week coming up next week, same time. On Thursday, where we're going to introduce a little bit more about R and pit tag data analysis and also introduce some other pit tag systems. So if you have fish that wander, like Ralph Lampman, if you're out there, your lampreys, you might meet some new friends and you can find your pit tag detections from some other place. So I am going to, let's see, make my slides advance, just a little bit of Teams reminders. The faces you see right now are going to introduce themselves. I'll introduce them in a minute, sorry. But for everyone else, we'd ask you just to mute yourself and have your camera off, just to help keep distractions down during the presentations. We'll use the chat for questions and answers, and also you can turn your camera on and unmute yourself later when we get to Q&A. I think everybody's got the Teams thing down by now. PNAP is a welcoming environment, and we're glad that you are all here. And I'm glad to be here today and to join you and to be able to host this great community. All right, so we have, this is a 2-hour session, if you recall, if you've been here before. We have 90 minutes of presentations, and then we have a 30-minute office hour at the end. So our 90-minute presentation is a team effort from folks at Columbia Basin Research. And you can see part 1, part 2. Part 1 will be about 60 minutes. Susannah and Matt Carter will present, and Jennifer Gosselin has been helping with preparing that work. They'll present and they'll have some Q&A, and then we'll take a moment, and then we'll transition to Rich Townsend, who'll present his work with Rebecca Buchanan in a separate session for about 30 minutes. Then we'll go to office hours. So stick around, plan to be here for the 2 hours. During that office hours, you're welcome to bring up any topic related to pit tag data, array analysis, tools and techniques and tips and whatnot. So for now, I would say, do you guys want to just take a quick introduction? Because we're not going to see all of you, but I know you're a big team. So I'm going to stop talking. We'll have you guys do a quick introduction, and then we'll turn it over to Susannah, I think, to kick it off. So maybe Susannah start and go around.
Yeah, so, well, it's at the beginning of my presentation, but I'm Susannah Iltis, the web computing specialist for CBR. Matt?
I'm Matt Carter. I'm the database manager at CBR.
Hi, I'm Jennifer Gosselin. I'm the senior research scientist and PI of DART at Columbia Basin Research.
Hi, I'm Rebecca Buchanan. I am a senior research scientist and co-director of CBR.
I'm Rich Townsend. I am a research consultant and have worked here for quite a while.
Awesome. Well, thanks for indulging me in the introductions. I'm sorry I didn't warn you I was going to do that, but thank you. It's nice to have faces with names. And I'm going to stop sharing, Susannah, and turn it over to you. Thank you so much.
Thank you. Okay, so good morning. I'm Susannah Iltis, the web computing specialist for Columbia Basin Research within the School of Aquatic and Fishery Sciences in the College of the Environment at the University of Washington. For this webinar, we will be presenting on Columbia Basin Research DART and tools specific to pit tag data. Sorry. I will be presenting part 1 with Matt Carter, database manager. I'll cover a brief introduction to Columbia Basin Research, CBR, including history and philosophy, and the Gosselin and Buchanan labs. Matt will cover the data access and real-time DART database, including Pitagus data handling and pit tag filters. We will pause for questions after Matt's presentation on the database and data handling, and then I will present on DART web-based pit tag queries. We expect to have about 10 to 15 minutes for questions and live demonstrations at the end of part 1. And so we invite you to submit questions in the Teams chat and take notes along the way. In part 2, Rich Townsend, Senior Biometrician, will cover CBR mark and recapture tools. And I'd like to note that the URLs referenced in this presentation will be provided afterwards. So 3 decades ago, Professors Anderson and Skowski founded Columbia Basin Research, CBR, to provide easy access to data, information, and analytical methods for in-season fish and river management and research. Today, led by Drs. Gosselin and Buchanan, we continue to strive to provide public access to data, value-added data products and visualizations, and statistical and modeling tools customized to meet the needs of users. While CBR is one research group and one website, is actually the collaboration of 2 labs. The Gosselin lab focuses on access to data, value-added data products and visualizations, and applied and mechanistic ecological research. The Buchanan lab focuses on statistical methods, mark recapture tagging studies, analytical software, and value-added analysis. In part 1, we'll be covering aspects of the DART database and queries from the Gosselin lab. Shown here is the data mega menu on the website, providing easy access to select DART products, and we'll dive more into this later. In part 2, Rich Townsend will be covering products from the Buchanan lab. Shown here is the tools mega menu on the website. And so now I will pass it over to Matt Carter to talk about the DART database.
Hello, I'm Matt Carter. I'm the database manager at Columbia Basin Research, and I work to obtain and process the data for our web products. To give a background on our Pitagus web products, we need to get into the backbone of DART, which is our database. We will start with the broad categories of other data on DART, and then we will talk about the concepts behind our database and where we source our data. Next, we will talk about the Pitagus-specific data retrieval and processing, and then discuss our PIT-specific filters, such as the life stage filter, the ESU/DPS filter, and juvenile transportation filter.
Sorry.
On DART, we host a variety of data to support the management of salmon and steelhead in the Columbia Basin. Broadly, the fish data we maintain are juvenile and adult salmon and steelhead, as well as some freshwater resident species. The environmental data that DART covers are river and ocean conditions, as well as some dam operation data. One thing that's important to note is that DART is a secondary data repository with centralized and integrated data from numerous other federal, tribal, state, governmental agencies, and other entities. I'll describe what DART is through this flowchart diagram going from data acquisition to preparation and then to the products and services that DART offers. We mirror data from many sources to aggregate and integrate data into a centralized location accessible to the public in real time. These sources include ocean and forecast data from NOAA, trap and adult passage data from various tribes, state agencies, and PUDs, water quality and dam operations data from the Army Corps of Engineers, river conditions data from USGS, various data sets shared on Fish Passage Center, and of course, pit tag data from PSMFC. Our database administrator, web specialists, and CBR researchers prepare the data for analysis in online projects. And these are created with as much foresight as possible and in collaboration with data owners and stakeholders. The products and services that DART provides are mainly archiving and integrating data sets, developing query tools for customized data downloads and data visualizations, as well as more specialized requests and other online predictions products. And we'll expand on these products relating to Patagas later in this presentation with Susannah. Now, we'll walk through a simplified overview of the Patagas data retrieval process. Daily data files are retrieved from the Patagas SFTP site, along with additional metadata or dimension tables that fill out the information of those data files. The daily files are split into mark, mortality, recapture, and detection files. Files are then loaded into temporary tables to begin processing. This process happens daily, and we perform regular upkeep and maintenance of the process. Data is then split into two separate tables, a mark/tag table and an observation table that combines mortality, recapture, and detection tables. These tables are then linked with the metadata tables to contain the full information for each observation event or each mark event. Data is then run through multiple data filters and functions to programmatically expand on the data, as well as aggregate tables by site, species, and observation year to speed up operations of data queries on the website and speed up internal processes of the data. Every optimization helps, since, as we all know, the Patagas dataset is very large and requires a lot of space to store all of it in one location. And if queries and tables are not efficient, it would take far too much time to run queries on the website. Now we'll talk about our PIT tag filters. We have three primary goals behind the creation and the maintenance of PIT tag filters. First, the filter must add value to the PIT tag dataset. Second, it must be data-based, meaning we should be using the data that's given to us to make determinations using as few assumptions as possible. Finally, we want to embrace the complexity of the river and Patagas system and try to incorporate exceptions when needed. For example, we cannot assume all fish that are detected in an adult ladder are adults, because hatchery juveniles are occasionally released above adult ladders, and we have seen juveniles that are out migrating being detected in adult ladder detections before. The filters we'll discuss today are the life stage filter, the ESU/DPS filter, and the juvenile transportation filter. For the life stage filter, there are three main categories of life stages: juvenile, adult, and unknown. The objective is to assign a life stage to each salmonid PIT tag detection. This will combine tagging, interrogation, and recapture datasets to determine the life stage at tagging and each subsequent observation. Again, this filter tool helps with additional analyses that we'll show later in this presentation. To create a life stage filter, we use Patagas data specifications to compare the tagging information to the detection information. Some examples of this include comparing the migration or release year to the observation year to see if enough time has passed that the fish is definitively no longer out migrating. Using the release site and release length, we're able to tighten those conditions as well to get a better sense of when a fish is likely an adult. We can also use detection sequences to determine if a fish is a returning adult, such as if a fish is observed in the Bonneville fish ladder and then a week or two later is observed at the Dalles within a certain time frame, then it is likely an upstream migrant. If we use a method to determine an observation of a fish is at the time an adult is in adult life stage at that observation, we can also use that to determine that all future observations of that fish are also assigned an adult life stage. The life stage filter is undergoing a revision to better streamline the processes that have accumulated over the last decade or so, as well as incorporating some improvements to detectors and dams and improvements to our systems as well. In a case of this would be using the order of coil detections along an adult ladder to see if the fish is moving up the ladder or down the ladder. Which is more difficult than you could think, because there's also cases of a fish trying to go up the ladder and then going down, but I digress. Oh, and to specify the definition, the DART definition of adult is a sexually mature upstream migrant, which includes precocious mini jacks and jacks and jills. The DART ESU/DPS filter identifies pit tag Columbia Basin evolutionarily significant units and distinct population segments listed for protection under the Endangered Species Act, based on pit tag specifications including species, run, rear type, and release location. In addition to those, DART also uses hydro unit, river kilometer, tagging coordinator, hatchery, and stock where applicable. We restrict ESU/DPS assignments to fish that are tagged as juveniles to avoid mislabeling adult migrants and strays. With each Patagus update, daily, monthly, annually, into DART, we maintain the database and report definitions of all the Columbia Basin ESU and DPS populations to match NOAA's stock definitions, including hatchery propagation efforts. Based on the aggregation and filtering of pit tag data, DART reports on the statuses and trends of Snake, Columbia, and Willamette River Chinook, and Sockeye ESU, and Steelhead DPS populations. This filter allows us to better aggregate tags and observations based on distinct ESU/DPS beyond aggregating just on species and run. Lastly, we have the Juvenile Fish Transportation Tool, which examines pit-tagged fish data at transport locations and subsequent detections to assign a probable transport outcome to each individual fish. For example, we could have a fish that may have been given a transport designation originally, but was actually detected at a downstream location, and thus was indeed not transported through the hydro system. The information used includes pit tag data and transportation information that is distributed from Fish Passage Center for the U.S. Army Corps of Engineers, as well as information on detectors and their configurations, such as information on whether a detection coil feeds to a bypass. This examination of data is conducted annually after the end of the transportation season, and we've been conducting this on data from the year 2000 through the present. And for dams, Lower Granite Dam, Little Goose Dam, Lower Monumental Dam, and McNary Dam up until transportation operations stopped there. Here we have a breakdown of the transportation statuses that we have assigned over the years, which shows that there's a large number of fish that could easily be misidentified by taking the initial transportation status for granted. As we can see, even though the majority of cases where a fish is identified as likely transported are correct, there are a significant number of fish that are detected at a bypass coil and/or are detected downstream after the initial transportation status. So to summarize, DART is a secondary data repository where we can centralize data sets for integration with web products, allowing us to add value to data. We covered how Patagus data is retrieved and processed, and as we've seen in some of the past talks in this series. And we also covered how we can create value-added filters for life stage, ESU and DPS, and transportation. We then use this data in our numerous DART data products, for which I'll be passing it back to Susannah. But first, are there any questions for this section of the presentation?
Thank you, Matt. Any questions? You can type it in the chat. You could come on and just speak your question. I see Ralph's
Um,
asking. Can you see the chat, Matt?
Yes, I can. So, is there any potential to include this filtering service for lamprey? I think so. I think there's potential, but we would need to learn more, and I'm not sure which filters. I guess it would be the life stage filter or the there's no transportation for lamprey, right? Okay. So I think there's potential in some of our filters.
Yeah, there's likely there might be they're trying to exclude them right now for transportation, but there's probably some that get inadvertently into those barges, and we're talking about potentially including them in the future, given the passage survival studies that we're starting to see. But yeah, that would be awesome. That's a
Yeah,
possibility.
If they get added to transportation, we would definitely expand the transportation filter to include them. I think
Yeah.
that's, oh yeah, Susannah might.
Oh, I was just going to say, given the way the filter is written, I think currently what we do is we focus on, Chinook, steelhead, sockeye, and coho. But the rules that apply for them would still apply for lamprey if they were ending up in the raceways and detected at the final transportation detectors, where I know there's a lot of collision. So that's just one thing to know, that we still have to rely on transportation operation dates, as well as fish or organisms being detected in the raceways, as well as those final coils before the barge, because there's not actually detections on the barge itself. But it sure seems like that would be expanding the analysis to other species is not that the same rules would apply.
Mm-hmm.
And like the duration of the their larval juvenile stage that could be set to based on the species, and it's flexible.
Yes, I'd say for Pacific lamprey, we would love to have more input for us to help us create the appropriate rules and logic, especially for different if they're for different parts of the basin or anything there. So we definitely appreciate any expertise.
Yeah.
Yeah, definitely. Happy to set that up.
Thanks, Ralph.
Yeah, thank you.
It looks like, is that Brian? Brian has a question.
Manshov? Go ahead, Brian. We can't hear you, Brian. Maybe you're muted. Or are you typing?
There you go.
There's
Okay,
a different
sorry.
question in the chat. After Brian, we'll get to you, Min.
Okay, so on the juvenile transportation filter on the table that you showed, am I interpreting that right, that it looks like about over 10% or so of the fish that are transported seem to be actually that are indicated for transport are actually not transported? Is there about 4 million 2000 to 20 to 24, so you have 4 million that are transported, and about over 400,000, almost 500,000 are transport but detected downstream or probable bypass. Am I looking at those numbers right?
Yes, and that has to do again with trying to combine all the different bits of information, so that we get the operations that are actually happening at the dam. So because you may be in the raceway, but then directed back out to river, and that's just part of the the operation. So it's trying to combine all of those. But so the first thing we assign is transport if it's seen at certain coils, and then operational rules are applied on top of that then change. So the transport starts at what coil. Like we've set a set of coils at a project means a certain outcome, and then operational information is applied on top of that. And so the ones that we say are, have that, probably transported would be your clean T. And then everything else after that, there are reasons either for operations that, they collected and then put back into the river, or there was actually, actual, dumping of a collection, and that would then end up being your detected downstream.
Mhm.
Awesome.
And I know that at least in some of the more recent barges, there's better pit tag detection capability on fish actually making it on the barge. Do you have any more recent numbers on that, or is that, is 2024 better, or?
We have not spent time necessarily digging into that question.
Okay.
So we haven't looked specifically at that. We do know that there is definitely coil tag collision with those final coils. But they add another point of information. And then so let's see.
As for the question in chat, I do think that that's probably a better question for the next section of the presentation, since that's more on the web itself.
Okay.
We're trying. That's the short answer.
Yeah. I guess it's not that hard of a question to answer.
But
You
a lot
have
of what,
about an hour to figure that out, and
Yeah,
we can come back
a
to
lot of
Min's
what
question later.
a lot of what we do, unfortunately, is big. So I will go ahead and jump into the queries, and thank you so much for the questions. We really appreciate that. So in the Dart queries section, I'll be covering the Dart queries purpose and design, the Dart web page briefly before moving on to the Dart pit tag queries and highlighting specific ones. So the purpose of Dart queries is to provide users access to data and visualizations, data downloads, summaries of relevant metrics, customized query interfaces, and user-centered tools. And for design, we follow these guidelines of attribution to data owners, transparency of methods, value-added services, and human-centered design. The Dart web page is organized into 3 main sections, starting with the Explore by Topic section shown here in the screen capture. It introduces users to select queries and products in 4 key areas. That's juvenile salmon, adult salmon, river ocean climate, and then integrated fish and environment queries. Also on the Dart web page are the sections for Explore by Query Style to dig further into our products, and the Access All Dart tools for the full list. So let's dive into some pit tag queries. The Data Meta menu provides quick access to select Dart queries, products, and resources. Here I've highlighted the pit tag queries specific to juvenile life stage based on the life stage filter. Next, we have queries and products for the adult life stage. And then we have the ESU and DPS specific queries and products based on the ESU and DPS filter. And you can find information on the population definitions in the metadata and glossary section in the resources list, and that's over in the first column. And then our resident species. To explore all the pit tag queries in full and other queries, follow the link to the Access All Dart Tools section of the Dart webpage. And if you don't find what you're looking for, please ask. So today I'll be starting with ESU and DPS graphics and text, and then adult returns, followed by conversion rate, SAR survivals, specialized analyses, and then I'll finish with the Dart version of the CJS and travel time estimates. So this is the query interface for the ESU and DPS graphics and text, which presents summarized detections as daily counts. I'd like to point out common elements found in many of the Dart queries, and that's the data source, related queries, output format selection, parameter selection, and date range. And then this query also includes an option for life stage selection. The graphic and text query style is the most flexible of the Dart queries, allowing for user selection of multiple years, multiple locations, multiple data parameters, and then offering multiple options for output format, including plot, HTML table, CSV, and then a tidy data format. And the graphics and text queries all share these features that you can customize the date range for like the x-axis of the graph. You can customize the data range, the y-axis, and plotting features like size, grid, and plot symbols that we'll see on the next slide. And the output HTML table is either by calendar date, that's month, day, or by day of year. And output CSV by multiple parameters per row, which is one row per date, or a single data point per row, which fits into the tidy data format. And then automatable customized data retrieval, and that's also on the next slide. So instead of submitting a request for a plot table or CSV file, a user can check the option to generate a reusable query URL based on the selections that have been made that then can be used for scripting automated retrieval. In the call out, I've included a short sample snippet of our code. Then there are additional customizations possible. Specific to this query, river data can be integrated with the fish data. Here, I've selected Lower Granite as the river location, and outflow and spill are also selected as the river data. And then fish can be customized for cumulative counts or normalized counts over the period. And then customizations for the graph include combined like data types, which allows plotting of outflow and spill, which are both in KCFS on the same axes. And then I've turned on plot symbols. And then for transparency of methods at the bottom of the query interface, we include query-specific notes. After submitting a query, you could see output similar to this. In this example, I have selected Snake River Spring Summer Chinook ESU observed at Lower Granite. That's both the bypass and spillway in 2025. And then I changed the date range to start from March 1st to the present for the juvenile life stage with outflow and spill measurements. And those are on the right axis, y-axis. And the fish as the primary data is on the left y-axis. The results include a CSV file for downloading of the data presented in the plot. And the plot includes a timestamp of creation for recording when the query was executed for the user's reference. So the next query highlight is a detail query style for adult returns detail by observation year. So based on the life stage filter, we offer an observation year detail report with selections for year, location, which may be one or more Patagus interrogation sites that we've combined into a location, species run, and rear type. Further refinement is possible with date range and optionally filtering the results by release location, which is either a whole water body or an individual release site. So similar to other detail pit tag queries offered by Dart, the results include a select number of tagging and release specific columns. So here we have tagging file, the number of fish released, tag ID, species run and rear type, length at tagging, if it was reported, the mark and release site, and the total RKM from the release to the ocean, and then the release date. I'll take this opportunity to point out the Dart specific extension that you may see on in Dart results, and that is the .dart and then a number that we have added to Patagus tagging files. And this was introduced in 2022 to facilitate Dart analytical processes that require unique release events. So we could see in rows 2, 3, and 4 in this table that those fish are all indicated. They're all coming from the same tagging file, but we have deemed them as actually having different characteristics. And that's probably mostly around the release date, defining them as to make them unique release events. And so going into that, we have coordinator, organization, marking method, release date, release site, release hydro unit, release river KM, hatchery, capture method, stock, brood year, migration year, and session message. The results include a select number of detection-specific columns, including observation site, that's the Patagus interrogation site, the first detection time, and the travel time since release. In this case, the location selected was Bonneville Adult Ladders, which we combine into one location with 4 possible observation sites in its current configuration with BO1 to BO4. So another adults return product offered is the historical run timing query style. This query style allows the user to compare timing of populations across years. Shown here is the query interface where the users can customize the request by species run rear type, observation location, and the number of the most recent historical years to include. And then there are refinements offered for release location, excluding mini jacks, date range, and then there's an option for multiple plots. This is the resulting historical run timing plot for Wild Spring Chinook, released at or above Lower Granite for the most recent 15 historical years at Bonneville Adult Fishways. So timing for the years is presented on the left with passage dates on the x-axis, and the number passing for each year is presented on the right. An HTML table and CSV file are also part of the results. One visible pattern we can see from the 50% date, which is the green vertical bar in each year in the plot, is that the timing over the last 8 years has shifted 1 week later than in prior years, and it's up to the user to ascribe meaning. So the conversion rate product uses information from the life stage filter and the transportation filter. The DART conversion rate metric is a simple calculation of the fish observed at the lower project with subsequent observation at the upper project, and there are no adjustments for harvest or other variables. The user can choose between the default results or one of the comparison options for species run rear type, reach, or in-river and transport. Here, the user selects the conversion reach, species run and rear type of interest, release location, and a month range of interest, and that's specific to the lower project. There are no restrictions on detection timing at the upper project, other than it has to be after detection at the lower. Further customization options are available for upstream detections, mini jacks, and transportation. So here are 2 output graphs produced from the product. The first, the left plot, is for the default results showing conversion rate, the orange-filled circles with 95% confidence interval on the left axis, and the annual weighted average arrival date, the gray-filled triangles, with the first and last dates as the bounds on the right axis. The second plot is produced by a comparison request for in-river, the orange-filled circles, and transport only, the blue-filled diamonds populations. And these plots are only part of the results. The results for comparison request includes a table of the data used in the plot. The results also include links to each of the separate analyses for further examination. So here we have the summary metrics for conversion rate for in-river and transport-only populations for each year, and then you would be able to look at each in-river and transport-only analyses in further detail. So the default request results includes individual summary tables for each observation year by release site, species run, and rear type combinations. And so that's the table snippet shown here for 2023. In this example request, I selected to include detections upstream of the upper project. That is, fish with subsequent detections upstream of the upper project, but not detected at the upper project itself will be included as converted for the reach. The summary row for Minam River releases detected as adults in 2013 indicates with the UPS and the additional detections type column that this detection type is present in the results. So from here, a user can follow the option to view detection details, the last column, to examine detection data by individual tags included in the analysis specific to each of the summary rows. This new request generates a detail table that includes each fish used in the calculations. In the resulting detection details table, the bottom table, the example shows that for fish number 4, the upstream detection date at WR1, which is the Wallowa River at RKM 14, is assigned to lower granite, and the fish is included as converted, and that's the Y in the last column. And then we note that the fish is not included in the conversion reach travel time calculations. So the next pit tag product highlight is the smolt to adult return SAR survival estimates. This product uses the pit tag filters for life stage, ESU, and DPS, and the transportation for analyses. In the query, there are different options available for what we refer to as SAR category, which defines the start location juvenile to the end location adult. For example, lower granite all to lower granite adult would be all juveniles detected at lower granite. That includes the bypass facility, those that are transported, and the spillway, and then assumed out to the ocean and back to lower granite as adults. The choices for SAR category include additional refinements for transported fish, in-river bypass fish, or all. So for fish population for this query, you can select from the ESU or DPS population of your choice. You can further refine the population by rear type, selecting to include all fish in the population definition, or the wild-only component, or the hatchery-only component. As well, there are additional options, including whether you'd like to exclude mini jacks and jacks. So with a request, you'd get a graph similar to this, and the data that make up these estimates are available in the data tables that are provided with the results. In this triple graph, looking at the bottom graph, the SAR survival estimates pooled by year are shown in percent for out-migration years 2000 through the present, as orange-filled circles with 95% conference interval. The gray horizontal line at 0.8% shows the 10-year average of the most recent completed returns for reference and comparison. In this case, this is 2011 through 2020. In the middle graph, there is the weighted average of the juvenile detection date, the filled triangles, with the middle 90% of the juvenile detection date range, which is the shaded band. And on the top graph, there are the juvenile and adult counts by age. The left axis shows the juvenile counts and the right axis shows the adult counts. So there are data tables that are available to dig into the data further and see the individual pit tags. And I will point out here, you may have noticed on the plots, the titles are detailed because these plots can be considered standalone products. And so all information that may usually be contained in a caption are specified in various places on the plot, including the timestamp of when the query was run and the product was produced. And then more detailed information is available on the web page as we strive to be transparent as possible and cite data sources. So this is the accompanying annual summary table included in the results for the data that are included in the plot. So you have the column for each of the years, showing what the juvenile weighted average arrival date was, the number of juvenile detections, as well as the adult detections, the SAR survival percent, the standard error, and then the adults are broken down and for individual counts for each of the ocean years. So here, because mini jacks and jacks were excluded, the youngest fish in this group are spent 2 years in the ocean. The results also include individual juvenile detection year summary tables by release basin with links to view the detection details. And this table for juvenile detection year 2021, and so for Snake River, Spring Summer Chinook ESU, the release basins include the main stem snake, the Buchanan, the Grand Ron, salmon, and Imnaha. And just to note, the Clearwater Basin is excluded from the ESU definition. And if we follow the link in the last column there to view detection details, additional results are generated. The requested detection details include fish-by-fish details for release site, species, run and rear type, tag ID, length if it's recorded, first detection at the juvenile site of the SAR request, and the first detection at the adult site of the request. And then summary metrics are specific to the year and the release basin are included. So for specialized analyses, I'll mention some of the options found on the DART webpage in the Access All DART Tools section. These analyses cover adult reach distribution, upper Columbia ladder fish movement, and species-specific detection probability at the fishways. And then for juveniles, we offer the lower granite bypass and spillway tables and the upload tag ID list analysis. And specific to spring spill period, we generate a quick look page with plots of the seasonal fallback rates for Chinook across years for each of the main stem and lower Snake River projects with adult fishways. And currently there is no user interface for the transportation outcome files we produce by year and species due to their size. They have to be separated. But if you're interested, please email us and we're happy to share. We're happy to get you connected with them. And that's web@cbr.washington.edu. And so I'll just touch briefly on the lower granite bypass. That's the GRJ and the spillway GRS tables. Here's the first summary table in the 2025 report showing outflow spill, number of spill hours, and then spill bay 1 hours. And spill bay 1 is the spillway associated with GRS, and then the spill bay 1 percent of spill information by date, and then the summary count by day for juvenile and adult space species. And so here I've highlighted with the shaded row, you can see on March 1, which was the first day of any spill at Lower Granite, that 12 adult steelhead were detected at GRS using spill bay 1 to move downstream. And then so adult steelhead continue to use spill bay 1 on an almost daily basis with a total of, at that point in time, 319 through May 8th. And then, so during March, there was a small bit of, the spill run through spill bay 1. And so you can see that in the highlight row that basically 100% of spill was run through spill bay 1. And then it shifts as we get up to that most recent date of May 8th, where it's 24 hours of spill, but the spill going through spill bay 1 is only accounts for 8% of the total spill at the project. And then the last highlight I will present is the DART pit tag release and observation summary for generating survival and travel time estimates with tag file selection, which I decided to call the longest query title in this slide. The query is the front end to configuring and submitting a survival or travel time estimates request through DART. This is a simplified variation of Pit Pro, which Rich Townsend will detail in part 2. Due to the complexity of this analysis, if you are not familiar or you need a refresher, we highly recommend reviewing the methods webpage. And so those URLs are provided there. So this is the starting query interface for step 1, which is define the fish group of interest. In this example, I am using the by release parameters to define my group. A virtual release is possible using the by observation option on the page. And I have selected release year 2024 with species sockeye and released in the Okanagan Basin based on release RKM. So the results from step 1 is a table of qualifying tag files based on the initial request. Additional tagging and release information is included in the table to assist the user in further selecting the fish of interest. And so that includes, having the tagging session message there, as well as, release RKM and release site and various other tag coordinator. And then in step 2, the user selects the tagging files of interest from this table and submits this intermediate query. So the results from step 2 produce a page of release and observation summaries particular to the group selected. The release and observation summaries are provided for the user to review, and I'm calling this step 3, but I'm not showing it here due to the length of what that looks like, before the user can either scroll or jump down the results page to get to step 4 to configure and submit a survival or travel time estimates request. In this example, I have selected from the predefined list the Upper Columbia through the main stem Columbia sites from the group list. You could select individual sites as well that have had observations. And then for estimates type, I've selected juvenile harmonic mean travel time. And due to the possible complexity of a request and time to execute, an email address is required for the delivery of results. So step 5 is receiving the results via email and a text file attachment. Here we see travel time estimate results containing 3 tables for travel time. The middle table is standard errors and the last table is counts, and each are presented by reach. And lastly, if you have a list of tag IDs you would like to submit for estimate analysis, you can do that too with the upload tag ID list product. And so to conclude, today we introduced you to CBR and our labs, provided details about the DART database and Patagas data handling and filtering, and covered a small portion of what we offer both for PIT tag queries and products, as well as other data sets on DART. And we invite you to explore at your leisure. And with that, I'd like to thank hundreds of people, especially Patagas and tagging projects, data collectors, our collaborators and users, and people who have worked on DART program at CBR in the last 3 decades. We also thank Bonneville Power Administration for funding the DART work. And so I will open it up to questions. Whether you have questions today or tomorrow, we want to hear from you. And we do really value your input and feedback. It helps make DART stronger and a better tool and resource for the region.
Well, thank you so much, Susannah and Matt and everyone. And Bonneville, it's good to acknowledge Bonneville's been funding this for a long time, and that's partly how we get to have such complex and thorough tools. So appreciate that. We do have some time for questions, and we're going to switch gears to part 2. So don't slip away yet. Hear more about tools in a minute. Any questions for Matt and Susannah? Also say Mari Williams just dropped links that were shared in the talk into the chat. And know that those links will also be sent to you in an email from PNAP next week when we do our little summary. And you can also access those links later on pnap.org at the event webpage. We can reshare them at the end of the 2 hours too, Susannah. So any questions? Looking at the chat, anyone? Any hands raised? Also have time for questions later. Russ?
Yeah, Jen, I just want to make a comment. Yeah, thank you again to the CBR team for putting the links to all the tools. You just did mention that we'll be posting those. I think one thing that we're trying to do from this workshop is to publish a list of tools. And so we'll get a survey after, and I think one of the questions we'll have is if you have additional tools yourself that you'd like to post and share. I think we're trying to create some collaboration page where it'll be easy to find a list of these tools, what they do, and links to them. So keep posted for that later.
Yes, absolutely, Russ. Thanks for the reminder. I do see Mike Ackerman has dropped a question in the chat. Can you guys see that, or do you want me to read it?
Yes, and I'm my brain is stumped.
For
I
the video
I have to think about that a bit.
I think that aside from the API request part of the question, I will say that we have had some issues sometimes with people programmatically pulling data from our website in terms of like overloading our systems. It's like too many requests happen at the same time. But we've been working on improving that, and we got a bigger server recently, which has helped with that, so that we can handle more requests from on the website of things. I think the only time we've run into a problem with that recently was when someone made sure that used R to make their request in parallel, which then hit us with a lot of requests from the same query at the same time, which overwhelmed our system a little bit. But yeah, so we are working on making our system work better for those programmatic requests coming through.
Thanks, Matt. And Mari reminded me that since the chat is not visible in the recording of this presentation, it is good to restate the question. So the question was about allowing juvenile and survival time queries via API request for our scripting. So you might have gathered that from the response, but I am restating it. And I think there's more thought going into that. I can see Susannah's you're thinking on the fly here. So.
Can you see the thought furrow between my eyebrows?
Yes.
Yeah, I'm trying to, so we've simplified it a bit on, through Dart, where we still need to, so we put the user through getting the release and observation summary so that we put it, that's that step to try, but we also have, I can't think of the word I want right now, checks in place so that you actually have enough observations. So, enough observation locations in the request. So that's part of making the user do it manually, is trying to get to the point that you're actually submitting a realistic request. So I guess in thinking about what that would look like with an API request, I'm unsure how to make what all would need there, but we can talk more.
Good fodder for thought. All right. Another quick question. Are we ready to switch? Keep in mind, you can save your questions for after part 2 presentation here. Anything burning, you just can't wait. Jennifer, are you trying to jump in? Sorry.
Yeah, no, that's fine. Just getting back to Mike's question, that'd be great to follow up on that and maybe run a pilot version and see what it takes and what might be some challenges. Maybe start small. And also for everyone else, it'd be interesting to hear if there's interest from others for this as well. And we have other staff as well that can help out with this and programming and our scripting, and we can do a trial run. So definitely we'll reach out to you, Mike. Thank you.
Yeah, sorry to distract on that one. I'll reach out to you guys separately. It's just something I've been curious about very recently. Appreciate it.
It's a good question. I think more and more we see data access through APIs so people can script, and would love to see you guys try that out and then share it back with the group later, like maybe late, not today, but another time. We're BNAM's anticipating more, whether it's webinars or workshops next fall, following on from this series. So we'll circle back and ask. All right, are we ready to shift gears to part 2? Okay. I see Rich's slides, so thank you. And remember, we'll have time for Q&A about this presentation, part 2, and then also the open office hours. Thanks, Rich.
Okay. Do you want me to go ahead and start?
Yes, please.
Oh, okay. I lost my picture of you, so I don't know what's going on. All right, so get my notes over here. Move that off to the side. My talk is going to be about the mark and recapture tools that we provide for fisheries and wildlife biologists to analyze their own data. I apologize. It's been a while since I've had to do one of these. So the introduction would be, I'm Rich Townsend. I specialize in mark recapture studies. I'm going to give a brief overview of the tools that we that are applicable to mark recapture study design. And then we'll go into depth on program PitPro, which is designed to process pit tag data into individual tag histories under defined assumptions. Information is needed.
Rich, I think you muted yourself or something happened.
Sorry, the little window keeps popping up and getting right in front of my view of the screen.
All right, go ahead. We missed a sentence or two, so maybe.
Okay, so beyond the overview of the mark recapture tools that we have on our website, it will go into more features about program PitPro, what input files are needed, what options are available to better fit your study, and there, I hit this button. The output options of reports that you have available to you. Again, I'm Rich Townsend. We've have over 30 years of estimating survival for fishery studies. Studies have used coded wire tags, balloon tags, sensor fish, pit tags, acoustic tags, and each of those have their own unique features that have to be accounted for when you're doing survival analysis. From these experiences, we've created flexible tools to estimate survival and enable biologists to better understand their data if they are actually doing the analysis. The data that for all these tools that we have on our website, those are available at our tools page. Along with user manuals, we have how-to videos and anything else and some example data so you can practice to see if you're understanding how these work. For mark recapture studies, our tools fall into two categories. Pre-study, where we're going to be estimating sample sizes, and post-study, which will analyze the mark recapture data. For the first of the sample size that we have is this program that will estimate optimal sample sizes based on to obtain the required precision for a number of study designs, ranging from a Ricker 2 release study with a single detection site to a single or paired release study with multiple detection sites. For more advanced study designs, there are three types of virtual release studies: a virtual paired release and full or reduced models for the virtual with dead tag release studies. The other sample size program we have is tributary survival sample size, which estimates sample sizes for a tributary survival study. Survival and variance equations are included in the user manual of appendices for those who are more curious about how we get these estimates. We have 5 programs that can be used for juvenile mark recapture data. A 6th program, Roster, includes detections from adults to estimate a life cycle model. SURF has a step-through process to estimate the main, group, and individual effects of survival detection and/or final lambda probabilities. This program allows us for a more advanced analysis. TribPit and Basin TribPit are expansions on the mark recapture design. TribPit analyzes the outcomes from a release site with multiple year detections for residual fish, while Basin TribPit is a more advanced version that can analyze multiple release groups simultaneously with possible common survival and movement probabilities to improve estimation. User is a flexible tool to estimate maximum likelihood parameters and provide estimates for functions using those parameters and their variances, which is very handy if you're trying to get like a pooled or a combination of survival estimates. PitPro was created specifically to process and analyze data from pit tag studies. As you can see that pit tag studies today will have hundreds and hundreds of, or even thousands of observations in the river. And this processes that data into a tag history by tag, and with additional information to allow you to document how you're coming up with these numbers. Roster includes detections from adults to estimate a life cycle model. An example of this is here on the right, where we're doing a 3 detection sites down 3 years in the ocean, up to 3 years in the ocean, and then 3 detection sites on the way back up. PitPro was designed to process the data from Patagus in a consistent and user-friendly program. The river and dam operations have changed through the years, or even within a year. So a site configuration file is used to document the conditions on the river. This file is regularly updated by DART and can be tailored to fit your analysis needs. To if it doesn't include everything, say you want to add a new pit array or change the outcome determination, you can download that and do that yourself. The program can also includes an error checking in the process, and you have the ability to save your settings and file names used in your analysis so that you can reproduce your results. So why use PitPro? A large number of pit tags are detected after death, for example, at avian colonies. The environment changes constantly due to nature, dam operations, fishing, predation, etc. To better analyze the detection data, it helps to know what routes are available at the time, whether transportation was occurring, where tags were recovered, etc. DART maintains an updated record of Columbia Basin systems configurations that PitPro uses to assign fates of individual tags according to provided assumptions. And again, there are user manuals, training videos, and example data available on our website. So this is the opening page for PitPro, where we try to setting it up for the initial analysis. On the top left of the window, tabs are in the general order of the steps you would take in analyzing the data, though you can use these tabs to move back and forth while setting up the program. Input and output directories are set up under the folders section. Oops, wrong one. And under the run configuration is where we decide what data to include in the analysis. The required files are the tag and interrogation files at a minimum. Optional are the recapture and mortality files, as these are used to remove or censor tags at locations based on whether a recapture event was something that may impact the survival of that fish. These 4 files have a standard report under or on Patagus that you can use to create them. Finally, under defined runs, this lists the runs that have been created but have yet to be processed. These can be output individually or to a single file containing all runs. And finally, the status box below shows the current progress of runs and notifications of any problems. The selection of desired output files occurs on the Files tab. The first 3 files provide the process capture history by tag, the reason a tag was removed and not included in the capture history, and the detection date and time at each detection site. The remaining 2 files are used by the program to estimate the travel time and how the program determined each tag's fate. This additional information can help you track down what may be causing results that you weren't expecting. The selection of the output file format gives you two options, SURF or roster format. These both create a tag history by tag with roster including adult detections. The covariates, the last section, has options to include covariates from the tag file for analysis using SURF, with options to remove tags with 0 and or null values in the analysis. On the next tab, the Sites tab is where the detection sites are selected for this analysis using drop-down menus. They automatically ordered in the assumed direction, downriver for juveniles and upriver for adults. There are two options available on this tab, a site release and a last history capture survival site. Site release indicates that the first detection site listed is a virtual release site. The last history site denotes that the final site that survival is estimated to. In a MARC recapture study, at least one detection site beyond the site to which survival is estimated is required, as you cannot separate the probabilities of survival to and detection at the very last site. The last site option allows you to create a pooled last site of multiple detection sites, potentially increasing the probability of detection below the last survival site. This file, or this tab for tag screening, allows you to create a single file of multiple species, run and rear type, and then filter them within the program if you want to download the data in fewer files. If you have already defined these values separately through your Patagus query, you can use these options as an additional check on your dataset. The Adult Settings tab helps us set up some important assumptions in regard to the life stage of each detection. Should we use it? And if so, where? The Assume All Fish Tagged to be Juvenile Observation does just that. If there are any adults in the data set, they will be censored if there is adult behavior. So it is up to the user to make sure that they really are only juveniles. At what point do you determine a fish to be an adult? The adult settings can be adjusted for a steelhead return year, or you can set a migration year directly, or you can set a juvenile cutoff date, at which point those tags were considered adults. Should we use mini jacks? You can either combine mini jacks with Ocean Age 1 class, or you can remove the mini jacks when they are detected heading back upriver. A little bit behind. How should adult fallback be handled? Last route indicates that when there are multiple upriver attempts, only the last passage is kept, while any route will credit a detection at a dam, no matter what order it occurred in the upriver direction. There are options to remove fish detected on juvenile sites as adults, or wait to remove adults at the juvenile sites after a set date. Additional options are available for dams with transportation operations. These dams may require more data processing due to the additional detections and routes. Oh, couldn't figure out why I wouldn't see anything. This data is mostly used for roster, though the censoring occurs at a transportation dam does occur for the juveniles as they're heading out. Individual routes outcomes of the dam are determined by a sequence of detections at the dam, and the detection history is either censored or continued down at that river. Transport dams that are not listed, tags are transported to those dams that are censored, and detections are ignored from that point on. There is an option to treat tags that have been sampled at these sites as transported. So using their detection history up to that point. The Advanced tab allows you to tweak how the outcome determination is determined. Removal trumps all means that any qualifying detection indicates it should be censored, even if detected downriver. While last detection, only the final detection at the dam will indicate outcome of the dam passage. The difference between removal trumps all and last detection is that there are situations where the detection route suggests that a fish was to be transported, but it was also detected on the return to river pipe. The first censors the tag at the dam. The second ignores the internal routing and determines that the tag was returned to the river in the end. Further investigation on the detected route and dam operations may be required, and PitPro can give you some help in that. Additional options are to ignore recaptures, which is useful where a tag was released and could be redetected, such as at a screw trap study that releases in a tributary. And then some miscellaneous operations. Allow single coil detections. This is a result from, in the early days of pit tag studies, a confirmed detection at a site required multiple detections to ensure that the tag ID and route were correct. As pit tag detection has improved through the years, this has become less of a concern. Allowing observed fish not found in the tag file is not recommended with the Cormac Jolly Seaver type of analysis, but it is useful if you are doing some data exploring and don't know what's in there. And finally, I'm really behind. The display warning messages may provide more information when you are having problems with the data. The navigation bar shows that is in the upper two rows of the program. These upper two rows are always visible and pertain to running the program, selecting the run configuration file, detailing how detections are to be processed, opening the results manager, or checking the program for updates. You can, under the File pull-down menu, you can run or cancel PitPro. And then the final three are load, run a configuration, or save the run configuration. A run configuration is the file used to save the settings of PitPro, runs, dams used, format of output, etc. So it makes it handy in that if you want to come back to an earlier configuration, you'll be able to easily set that up. The site drop-down menu allows you to select specific site configuration, or check updates for a configuration file, or download a copy to edit. The configuration file is, I have an example off here to the right. This is what documents what the river configuration was through time, where you have you can see the date of the last time it was updated. There's the codes for the different detection sites, and then outcome for within, say, a dam or area, what the outcome should be for tags going through there. So after you've hit the cog and processed your data and you've gotten your results, a new window pops up called the Results Manager. The summary reports here that I'm showing show the 5 files that are possible, though only the capture history, detection dates, and error files are required. The sequence and travel time files provide further documentation of the data processing. These are viewable through PitPro with active links or independently as a text file in the output directory. The data input file records your files that you used for this analysis, and these are active links as well, so you can open those up directly if you want to go back and forth and see what's going on. The error report lists the number of errors found in the report and removed by error category. In this example, you'll see that there are 22 tags that were observed going out of sequence, which meant going in the wrong direction assumed for a juvenile, and 27 tags that were removed before their first detection site. To further investigate what tags were removed and perhaps why, you can click on the Summary tab for a list of detection IDs and errors for which it was flagged for. This highlighted tag, for example, indicates that column 7, observations out of sequence, which was the reason that it was removed. To go further into that detail of why that was given an out of sequence error, you can go to the PIT tag tracker tab, which lists the detections in detected order with site, obs, the date, and determined outcome, and detector name and coil and source file. Here we see that the juvenile tag that was detected at Ice Harbor after 2 months after passing Bonneville Dam in an outbound direction. Thus, it gets an observation out of sequence error for going the wrong way. Other useful summaries provided are the arithmetic and harmonic travel times means. We provide the harmonic means as an alternative option as it is less susceptible to extreme outliers than the arithmetic means, especially with small sample sizes. The counts at the bottom provide a quick summary of how many tags were observed and where. Finally, the Cormack-Jolly-Sieber estimates are available in a couple formats: the multiple rows or one row, with or without the final survival to and detection at the last site. And these can be saved as CSV files. We are in the process of updating PitPro to include avian colony pit tags detected below Bonneville Dam and providing the user the ability to define the adult return year to better align with unique species migration behavior. Any questions?
All right. Thank you, Rich. I'm here. I'll turn my camera on and you can see me this time. Excellent. Wow, lots of stuff. There was a question in the chat about accessing files on the CBR site from Richard Hogg, but I think Susannah has answered that. Are there any other questions for Rich or Rebecca? Team here working on all these tools. Get the chat again.
Shock and awe.
Right. Shock and awe. It is a lot of details.
The thing I do want to emphasize is that our website, though I tried to rush through, I didn't mean to rush too much, but we do provide a lot of training on our website with videos that walk you through it and example data that's available so you can practice to see if you're understanding what is the process.
That's a really good reminder, Rich. I think if you saw something today that you're enticed by, but you feel like you didn't get all the details, or in a week or two you've forgotten them, remember that you can go to the CBR website and look for those training materials. It's really cool. And I see that Susannah has added into the chat about a reminder of the connection to Patagus, and that it does take a couple days for updating information across. So I think that's a useful thing for people to remember too. We're so demanding now. It's amazing, these tools, like they weren't even possible. I don't want to date myself, but you know, it's been a few decades. This access wasn't even possible, you know, 20 years ago, and now we want it like that. So give it a couple days to think about the connection between Patagus systems and CBR.
Well, so for that, we are the next day. So
Good.
so for
Okay, good.
for data, we are loading, you know, our most recent day, unless there's actual failure in the load, is yesterday. So we load, you know, that is happening every morning, and then there's updates. So we are staying synchronized. As far as the site's configuration, that was where I was saying, because there is a manual part to maintaining the site's configuration. So when we get notification, when Patagus has updated their site's configuration, indicating, you know, we track that, and when we pick up something new, then an individual, generally me, comes along and updates the site's configuration file that we use for multiple tools that Rich just detailed here. And it also feeds into the Dart simplified version of the CGS and travel time. And I happen to have updated it this morning and potentially confused an individual. So my apologies, but yes. So we don't keep prior versions. So what's available at that URL, the URL for the site's config, is always the most recent version.
Well, one of the nice things about that file is that it has the history of the interpretation of the different coils and antennas at each site. So it doesn't just have the current definitions of them. If they have changed through time, then the site's configuration includes that too, which allows you to then use that for historical data processing, and means that you don't have to go and look up how to interpret all of those coil IDs and track the tag as it moves through a dam, for example.
Which is really important when, as we go through time and just keep compiling up the years, you really don't want to have to go back, you know, decades to figure out what was going on.
Right. Very cool. Any other questions out there? I think Min Doan had to slip out, but he did have a question we didn't really get to, and I will be talking to him soon, so I could relay your answer. He was asking about web access to the tools, right?
Right. Well, with the handheld devices or tablets, you know,
Yeah,
basically smaller than a desktop. So we definitely have in our most recent website, which we revamp, organization, update that we did about, now I'm forgetting, it was a year ago, 2 years ago. So I think it was two. We did add a fair amount of code to make it my words are missing. Device responsive. That's the word I wanted. Still, I would say a lot of our data products and tables, when we produce detailed tables, they are quite large, and so viewing, I think, is nicer on a laptop or a desktop monitor versus the phone, but everything is available on the phone. It just may not be, you know, it's not necessarily designed for that. And then the other part of the question was whether you'd also need constant internet connection, and that would be, in general, yes, for our products.
Very cool. Thank you. I think those are, I mean, I wish you were still here, but I think you might have been thinking about, you know, where is that app in the whatever the app store to download so that I could just have all this stuff on my phone all the time. And I think someone mentioned how big these data sets are, and also you guys have a lot of tools, so it would be a lot of work. Min is doing some interesting things with Canada DFO, and so we've been trying to think about how things might be adaptable for their uses. So I'm glad he was able to join. All right. I'm filling the air with banter. Are there any other questions? I am not an expert in this, so I've learned a bunch today. I don't have any good detailed questions. Anybody else? We can also shift gears to the office hours. I think I want to remind everyone before we do that, that I want to thank the 5 folks who've been on the call today who were part of the presentations. You had 3 speakers, but there's 5 people here. Oh, Brian has a question. Thank you, Brian. Go ahead. You're muted if you're talking. Can't hear you.
Always fun to do that.
You're just teasing us.
Going back to the filters, and specifically interested in the life stage filter, and in pit tags in general, I mean, a life stage, you can think about assigning a life stage to a given observation. What is the life stage of that fish, and there's lots of different clues which can kind of tell you, okay, where what is that, depending on prior or post detections of that fish. But in the end, it's very probabilistic. I mean, you have an idea of what it is, but you're not sure. And so in terms of what DART does, is that, would you describe it as more deterministic, that you come up with some kind of set rules that says, okay, if it's this, almost like a decision tree, or is it a little bit more involved in that, in terms of looking at a probabilistic model where you assign the most probable life stage at a given detection?
The goal is certainly to make it as deterministic as possible, and that is part of what we're also trying to do with the update, is trying to create a set of rules where we are having a like a flow chart, like flow tree, and like the main basis for it is like you start with the assumption that whoever tagged it gives it a life stage on tagging that is in that tag file. And so we start with that, and then we go from there. So if a fish is tagged as an adult, then yay, it's an adult. Every observation of it is an adult. And then otherwise, if it starts as a juvenile, we start with that assumption, like, okay, all of the tags are juveniles, until we find like different reasons that we can say, okay, now we know that this is an adult because of whatever we decided are those rules. And so we're still working on a newer version where we'll have, and part of that goal will be to also share those rules, so we have like that laid out and better documentation for what exactly the rules are. Yeah, but yes, the goal is deterministic, not probabilistic. So like if you're doing probabilistic, you can probably say, oh yeah, all of the fish in the adult ladders are adults, because I would assume a majority of the fish in the adult ladders are adults, but that doesn't mean that there's not many exceptions that we need to make sure to take into account.
Yeah, I mean, thinking more like the transportation filters is another, you do have sort of somewhat of a probabilistic spread there.
Yes.
You have different categories of, you transported, it was detected here. But in the end, I mean, you've got the information is kind of sparse in terms of ruling something out for being transported, because the detection probability, if you're detected at lower granite, the probability of being detected again is pretty low, even if you were not transported. And so come up with some sort of an estimate of how many you miss, or how many you misclassify as being transported that weren't. But in the end, that's probably an underestimate, because you can only rule it out if it was detected again, and that doesn't always happen. And so I guess, being able to, assign more of a probability to that, gives you some way of adjusting, if you're looking for an effective transport, or at least being able to quantify, okay, um, you know, we know that this fish was not transported, or was transported with such and such a probability. I mean, it's kind of hard to come up with an exact number, but thinking more beyond just a binary transported yes, no, or and that's
Adult or juvenile.
yeah, and in the same way, with adults, I mean, there's different, different sort of classifications of life stage because, if you see a fish coming down, is it, or if the fish being detected in an adult ladder, I mean, is it, is it, if you can, is it really going downstream? I mean, this juvenile, is it a mini jack coming back, or, there's lots of different subcategories within each of these things of what an adult is doing, and that all depends on knowing all the other detections. So just sort of rambling on, but I mean, it gets really complicated to think about, being able to really know what a fish is doing at any given time. And so is Dart thinking, kind of, further down and different ways of trying to either both classify what life stages are, or and make that available in some sense for, through an API. So, you have a list of fish or a detection of a given fish, and you want to know what it's doing at that particular life stage. Is that something that's possible, or is it too complicated to do in real time?
Well, thanks, Brian, for your question. How about I ramble with you? We've thought about how much we would QC the data, and especially with pit tag data, different life histories, it can get very complex. There's so many different possible exceptions when you get down to the biology, the life history of different species and runs and rear types and such. And I think with a lot of other data, even environmental data, we thought about to what extent do we QC the data? And we try to minimize as much as possible to stay close to what's being reported, but correcting things that are pretty obvious that it needs QCing. And we thought about having different versions of data sets where do you have the raw form or minimal QC, or here are a bunch of other rules that we've applied in a different level or grade of QC data. We've thought about it. We haven't gotten to a point of doing it further, and that might be part of version 2.0. And we've reached out to a few folks already to look through some of our rules and to get some feedback, and we'd be happy to chat more too, Brian. Is that some good rambling with you?
Well, that's great.
And I guess what I would say too is that we're definitely not going to ever claim that these filters are like 100% guarantee this fish was transported, 100% guarantee this fish was an adult at this. Like, these are our best attempts to add value and make our best determinations of what we think of what using the data we can assign these values to. So it's we will make no claims that and I think that going back to what you were saying about the probabilistic determinations, I also think that is interesting, and I think that could be a really good project for us to work on. It's also very hard to figure out exactly okay, what is the probability that it missed the bypass or missed all the detections going downstream. There are things we can do about that, but I don't know. It's complicated. It's very complicated. So
Matt, I'll add on to what you were saying about applying a rule and getting a sense of how much improvement. We've kind of looked at that, and I don't think that I agree. We won't know the truth exactly of each detection and what life stage they were at, but we try to get a sense of, okay, if we applied this rule, how much error did we possibly eliminate? So for things that we are just sure about that is an error, once we apply that rule, Matt, I don't remember some of the rules and the percentages of sort of correction, but we try to aim for rules that do improve the data set by quite a bit. And at some point, we just go around in circles, perhaps, that we apply very, very specific rules and there's minimal improvement or we're not sure it really did improve.
Yeah, for a little bit of background into the process of the adult filter, or like the stage filter 2.0, what we're doing is we're taking like a year subset of the data to look at like a smaller subset to start, so that we can actually get to know the fish that we are applying this filter to. Not like all of the fish, obviously, it's still a very large number of fish, but we can see and go through and check on individual tags and like look at their detection history and say, okay, did our filter using our human eyes that this is the where we can see, like, using our human eyes, that this is an adult at this point, because we're better at that pattern recognition than computers still. And then we're using that and then trying to make our rules fill those, while also looking at the fish that the rules catches and making sure we don't get those false positives either. So it is a balancing game, and it's taken us longer than we expected it to, because it's impossible to necessarily, like, you can't get all the rules down to the point where you've gotten every exception to go the way you want it to. So but we're doing our best, and yeah, we do, like, we'll be looking for more feedback on that as well, and what rules are important. And yeah, we've been looking into probabilistically, like how much we can estimate our error is, but it is hard to estimate that. But yeah, that's a good thing to think about.
Very cool. Thanks for the question and the answers. There's always something else to figure out. I'm glad you guys have already been working on this. It's complicated, clearly. And I don't want to disrupt questions, but I am going to flash up the promo slide for the next week. To remind people, just a quick break. Next week is the 5th and final week of our 5-week series. We have speakers from USGS who are going to do some other applying R to pit tag data, and I think it's has to do with amphibians, if I'm correct. Kind of excited about that. And we're also going to have some sort of teaser short lightning talks from 3 systems that are outside the Columbia Basin that are using pit tags. And those are, I hope that's helpful for folks who've been asking about, you know, can I potentially find tag information from some other place besides the Columbia? So we're going to tee that up for next week, same time, same place. And you'll get an email following this one on next week saying here's how to access the materials from week 4, and don't forget about week 5. So we thank those presenters in advance. We really thank the presenters today. And while you've been listening to my commercial break, are there any more questions for our speakers today, or anything you'd like to pose building up to today? I really like Ben Staten had a question early on for a different presenter about what was your workaround. So it's interesting when people share their lessons learned and their workarounds might help someone else. Don't be shy to jump in here. So I can go to the office hours slide. And if you don't recognize this location, this is the headwaters of the Columbia River. It is a very small spring in British Columbia, and I recommend going there. It's really cool. It was quite serene, actually. So, anyone with questions? Anyone else on our presenter team from today want to respond to any of the previous questions? I didn't mean to cut you off. Right. I'm glad I got my photo up there. That was my vacation last summer, so squeeze that in at the last minute. Anybody else? Hi, Russ.
No
Question,
questions. No, just, nope,
comments?
just, I think we've hit the end. Thank you very much. I just want to say thank you for everybody. So,
Thanks everybody who came. We had another 65, 70 people at the start. I see people dropping off in the last 15 minutes. So it's getting to be lunchtime, perhaps. Thank you all. We'll get the recording up to the YouTube channel and you'll see some notes and all those links again. There's a ton of links, and don't forget that there's help at those links as well. So if you are thinking you didn't catch it all today in this whirlwind of details, you can go to the CBR web pages and look for those training videos and help materials, and also the humans. So thank you, humans, for being here today, and we'll see you next week.